What's up, you guys? Rex here. So I finished another week of medical school and we are continuing to learn about the brain. And this week especially, I learned some absolutely amazing stuff. So it's really hard for me to pick one thing, and so it'll probably be a couple things to share with you guys about the brain that I learned in this week of medical school. So I want to share with you a little bit about how plasticity works in the brain, specifically long-term potentiation, long-term depression, and spike timing dependent plasticity. So to keep this video as brief as possible, I'll probably be making quite a few assumptions that you have some general understanding of how action potentials work and how synapses work in the brain, but we're going to try and make it as simple as possible. So when we talk about plasticity, that basically is the brain's ability to change its circuitry and physiology in some way. And so if we think about, in order to form a memory, there has to be some physical change that occurs in our brain so that is now stored long term. And this has to be stored maybe for a couple minutes or stored for years. We remember things from our childhood even when we're 80 years old. So there is some interesting change that occurs in our brain. And we don't know, to my knowledge, exactly how this works. I especially don't know how it works, but I don't think anyone knows 100% how this works. But we do understand some mechanisms of how plasticity occurs in the brain, such that there are physical changes that would be related to how we are encoding memory. And so one of those changes is long-term potentiation and long-term depression. And so essentially the goal here is that it acts only on a single synapse. And what happens is with long-term potentiation, if an axon sends a signal, it is more likely to get a response in the receiving neuron. And so that is that it has an increased excitatory postsynaptic potential. While with long-term depression, there is a decreased excitatory postsynaptic potential. And so when this happens, even if we're sending signals from an axon to neuron number 2, because it is in this state of long-term depression, it isn't as likely to carry on that signal and shoot off an action potential itself. And so it was discovered that you could sort of artificially cause long-term potentiation by doing very high frequency action potential stimuli in neuron 1 moving to neuron 2. And so then this synapse that is connecting neuron 1 to neuron 2 can become long-term potentiated. Versus if you do sort of artificial low frequency stimulation, you can have where it's going from neuron 1 to neuron 2, now you have long-term depression which occurs. And so now this neuron number 2, even if neuron number 1 is trying to send a signal to it, it's never going to fire as a result. And so that is one way that the brain changes itself. Now, it's really cool that we do have some understanding on a cellular level of how this works. And so this works where if I draw a really ugly synapse, and so we'll say this is the axon, so this is neuron 1, and this is part of neuron 2, this is the dendrite. So we have some understanding of how this works, and I will draw the ugliest little receptor here, and we will call this an AMPA receptor. And then I will draw the ugliest receptor over here, and we will call this one the NMDA receptor, NMDA receptor. And so when an action potential reaches its way going down this axon, and what it does is it signals it to release glutamate. And so we'll just draw some little gold dots and we'll call them glutamate. And so they hook on to the receptors, this AMPA receptor and this NMDA receptor. So these receptors are actually ion channels that are activated by glutamate. When they receive glutamate, now they open. And so this AMPA receptor, when it activates, it allows sodium ions to flow through. And when you have sodium ions flowing through, this is causing an increase in the membrane potential, getting closer to threshold and trying to be an excitatory postsynaptic potential and trying to get this to fire an action potential of its own. Now, this NMDA receptor works similarly, that when it has glutamate attached to it, it tries to activate. However, it also has this little magnesium ion that sort of serves as a quark that blocks it. However, it only stays there when the membrane potential is relatively low, relatively hyperpolarized. So if I sort of just very basically draw out what this looks like, is that we have some resting membrane potential, maybe around 80 millivolts. When glutamate binds to the AMPA receptor, it's going to cause a little bit of spike. It's also bound to the NMDA receptor. However, this isn't having any contribution yet, but maybe at like this point here, this is when the magnesium is kicked off, and so now magnesium is able to get pushed away and it doesn't exist anymore in as far as blocking the receptor. And once we reach this point of threshold, we can sort of ignore this magnesium because it is no longer there and it gets pushed away. And once that magnesium is removed, we can actually have two ions that flow through. We have the sodium, which flows through, but we also have some calcium that flows through. And now this calcium is very significant because this calcium, it goes through and it activates a bunch of secondary messengers and it does stuff and this and that and this and that. Not super important for this video, but the end result of this messenger cascade started by calcium is that if there is a lot of calcium, it actually leads to having an increased number of AMPA receptors added to the membrane of this dendrite. So now the next time that there's a little bit of glutamate, there's going to be a greater amount of sodium that rushes in. And so that case is where it is long-term potentiated. And it's really interesting that if there is sort of a low level of calcium that is brought into the cell, in the case of having low frequency stimulation, the opposite effect happens where all of the AMPA receptors are brought inside the cell and no longer on the membrane. And so in that case, you would have long-term depression. And so that is something that happens in sort of an artificial environment where you have this really nice high frequency stimulation that you can do in a lab and low frequency stimulation that you can also do in a lab that isn't exactly something that happens physiologically. But we also have a similar mechanism that works in a real physiological sense. And this is something called spike timing dependent plasticity. And so this is something super interesting. And it is where if we have a axon that fires, and so we have cell 1 and it's going to cell 2. If we have an axon that fires just before cell 2 signals an axon, it is more likely to be long-term potentiated. If it happens, so this is happening just before. And so that is essentially that if it sort of has what seems to be a causal relationship, that neuron 1 fires and then neuron 2 fires, the brain says, oh, that must be connected. We should increase the likelihood that that happens, long-term potentiation. Conversely, if neuron 1 fires just after neuron 2 fires, then in that case, long-term depression occurs. And so we can plot, we'll say that this is time before neuron 2 fires. And so here we'll have, this is positive numbers and this is negative numbers. And so a positive time before means that it is happening before neuron 2 fires. So time 1 is before 2. This is 1 before 2. And this is the opposite, where 2 is before 1, where we have negative values. And we can draw our y-axis here, and we will say that this is like the relative excitability. And so we'll just call this like relative percent excitatory postsynaptic potential. And so this is compared to our baseline, how much more excited neuron 2 gets when it receives a signal from neuron 1, or how much less excited. And so if we just draw it and we say that baseline, this is 100% excitability, so anywhere down here is less excited, anywhere up here is more excited, we get sort of a distribution that looks like this. Such that if we have a neuron that fires right before, in this area, it increases the excitability of it. And if we have a neuron that fires just after, such that 2 fires its signal actually before 1 did, we actually have this decrease in the excitability going forward. And this effect can last for hours, days, potentially months. We don't know exactly how long it lasts because it's difficult to study that in like a mice model that doesn't live for 2 decades, and that's also a really long experiment to run for 2 decades. And so that's generally one example of how plasticity works in the brain. And that is just probably a small part, but you can imagine how rapid this can happen. And so if I go back to, I think it was last week's video where I talked about cerebellar reprogramming, potentially what was happening was inside the brain, there were synapses that were long-term potentiated such that these were more correct in helping you throw to the target. But as soon as the brain started figuring out those weren't working correctly, it started to long-term depress those synaptic connections such that it long-term potentiated other synaptic connections. And so how this all gets put together is well beyond me, but just seeing this little bitty sliver is amazing and sort of hinting at the total complexity of the brain. And it's really cool to learn about, maybe even too cool to learn. And it's really cool to learn about, even if it's something I'm not super interested in spending my career dedicated to. But I hope you learned something new. If you have any questions, comments, or concerns, I'd love to hear about them down below. As always, like the video if you like the video, dislike the video if you dislike the video. Make sure you subscribe, hit the notification bell if you want to catch my future uploads. And until next time, don't be ordinary, go be great.